what I want to introduce is, of course, the topic. What do we mean with those words, smart microscopy? And also what I will show you is like what is much more interesting for me is offering smart microscopy as a service in an imaging facility, and what is that might be different from what you would do on a research group, right? So first of all, just to let you know where I come from. So I am a scientific officer at the Center for Cellular Imaging in Gothenburg, Sweden. We are a national core facility that integrates both light and electron microscopy. And we offer open access to this equipment to any researcher that might be interested. Those include also companies as well, and they might come from Sweden or from abroad. Depending where you come from, you might have to access in a slightly different way. For example, nationally via the national microscopy infrastructure, or if you're in Europe, basically via Euro-BioImaging, because we are part of the Swedish node of Euro-BioImaging. The center has a wide range of services. We go all the way from wide field to super resolution like microscopy. And we also are heavily invested in 3D electron microscopy with the scanning electron microscope and the transmission electron microscope. And recently, we also have a pilot service on MALDI imaging mass spectrometry. And of course, as you might see, we are really a multimodal facility. So the thing that I like the most, that's the greatest fun for me, is to actually start to correlate all of these imaging modalities together. And of course, then that brings the topic of image analysis, right? So once we have gone through with the same sample through different methods, then we want to put them all together to create a holistic view of what we can learn from the different methods. What you see at the bottom is all my colleagues that make all these efforts possible. And then we have many different complementary skills. So if you are interested, please go to that website and you will find more information about us. Now, the plan for today, as it was said, is basically first I will start with a general introduction to the topic. So I hope that after that we are more or less all agree on what we are going to be talking about. Then I will take the opportunity also to show some general application examples. These are some pilot studies that we are running at the facility, and the idea is that we believe by developing these pilot studies that then we will also be able to develop more broad range services to our users. And then I will close up with some comments on workflow design. So if at any point you have any questions, just please feel free to interrupt me, and then I will keep track of time and everything. So don't worry, you can ask questions during the presentation. So if we are here today, it's also because we believe, or we are extremely interested in what I would call image-based quantitative biology, right? So the idea now is that, of course, imaging has been used in many different fields, but within biology, what we are sort of believing in is that once we take a biological image of our sample, there's a rich amount of information in those images. For example, I show at the bottom a paper from 2015 where they managed to create this sort of model for, let's say, a healthy cell line versus a diseased cell line. And then when you use stainings, which are not necessarily even specific to a particular target, because they didn't know what was going to happen, we would immediately see the difference, right? And the idea is that because we see those differences by eyes, we should be able to calculate descriptors from those images, which will then allow us to then achieve meaningful results in a cohort of images. So the problem or the challenge in quantitative biology then is going to be that many samples have to be imaged in order to achieve statistically significant results, right? So because of that need of imaging many samples, we have moved to study biological systems that are also inherently complex. And that brings, of course, we might have many different cell model organisms, but even within a single model organism, we might have, like cell differentiation, and also within a cell line, then we have the cell cycle, right? So there is a lot of complexity, and in many cases, the biological observation that we're interested in happens in a very specific subset of phenotypes of interest, which are very rare. And therefore, we have to sort of look for them in a sea of perhaps not so useful data, right? So again, it's not only that we have to cover a lot of different conditions, but even within one condition, we might have to image a very large area to be able to find what we want. To answer this question, within the field, we, of course, this is not new. We have been doing a lot of what I would call automated microscopy, right? So the idea in this case is that we are going to have a microscope to which we can interface with a computer, with software, and we have the capacity of controlling different parts of that microscope. And what I show here is basically an example coming from high content screening. And then in this sort of classical sense, you will have, okay, let's say a multi-well chamber plate or any sort of other arrangements where we have cells in many different conditions. And of course, we don't want to go there manually well by well, right? So now we can program our microscopes to go one well at a time and then adjust for differences in the wells, but at the same time, sort of do a workflow where, for example, what I'm trying to show here at the top is, let's say, if you look from the macro scale, you might see already obvious differences between the different wells. But then when we zoom in, we can start to see also at the well level that is not homogeneous. And then if we zoom even more in, then in many cases, we also can achieve single cell resolution, right? So we could analyze this data at any level, right? And this is, again, a very, very rich data set. However, in the classical sense, this is not what we would call smart microscopy. And the reason why I say this is that usually the microscope is programmed to follow a specific acquisition workflow, right? So the idea is that the microscope knows what to do for each well. It has this sort of protocol, this recipe, like a cooking recipe, and it's going to do it each well at a time. Of course, it's adjusting for differences, but it's not changing its behavior on the fly, right? So what we are going to now call smart microscopy is something which is slightly different. So there has been, of course, much effort over the last, let's say, 10 years to connect bioimage analysis workflows with these fully motorized and computer-controlled microscopes to generate what we would call automated and adaptive imaging workflows, right? So now the difference is going to be that, for example, if we were to see a classical picture, would be the picture here on the left, where we would have, for example, a motorized microscope, which we connect with some sort of interface to our computer. And that, of course, can be the nice software that the company that provides, but also perhaps if we need something slightly different, we might want to go to some open access, or if you have built, I was in the field where we were building our own microscopes. So maybe, you take a body from a company, camera from another company, and now you need some extra, third-party software to be able to connect them all together, right? Such a micromanager or micromanager. Now, this part would be, again, like let's say the strategy of the classical high content screening, right? So I can control it, I use it. Now, at the same time, we have done a lot of effort on the other side, which is the image analysis side, where now we have beautiful advanced models for segmentation, object detection, image-to-image translation, restoration. This field is booming very quickly. Now, what smart microscopy tries to do is that it tries to connect those two worlds together, right, via the software. So what we can do is that we can take an image of the sample, then we can try to find a particular biological target, or we can even check if it's there or if we have to wait, right? And then the moment something interesting is happening, we might tell the microscope, okay, now please go and change your behavior, right? So the idea is that there is this sort of feedback loop built into the pipeline, and that would change what I would call automated microscopy to what I would call more smart microscopy. All right. Any questions there? No? Perfect. So now, as you will see, one of the things that is going to quickly happen if we are successful with this is that, in my opinion, smart microscopy also must include strategies for handling the data that we are generating, because we can easily start to create, terabytes of data sets very quickly. And due to the size, simple data operations that before you could just do in your laptop or in your computer are not possible anymore, right? And this challenge of big image data is being tackled on many, many different fronts. So then there is, of course, the development of next generation file formats. We have, of course, new visualization and software tools and so on. And then the idea is that, of course, what we want, especially at the facility, is that we don't want just to have give access to the user to, okay, this is a microscope, this is the pipeline, you get all the data, and then, have a nice day. We also want to help them to manage this data. And in helping this beautiful illustration here of, from that sea of data, help them to store it, arrange it, and then finally put it in a nice story in the article once they have understood the biology hiding behind. And we really want to go through all of this without the users or myself feeling like my little friend on the right? So we want to really help them as much as possible. Now, there's another thing which I find super interesting. And of course, for example, in the field of light sheet, this is kind of obvious, but in some fields, perhaps not so much, is that once you post-process your data, that data could be order of magnitude smaller than your raw data, right? And if you are kind of happy with that, you might kind of save a lot of resources from your IT infrastructure in terms of, servers, transfer, IT infrastructure, and so on, because now you don't have to necessarily work with the raw data. Perhaps you can have this automated post-processing built into the software, which then makes everything afterwards much easier, right? And this is, of course, one of the topics that we will cover, I think, in session number 3, right? Now, from my point of view, then what I'm very, very interesting is smart microscopy as a service in a core facility. Okay, so why I think this might be slightly different is that if you are in a research group and you have a very niche application, you work really, really hard to make the best possible workflow for your scientific question. And these sometimes end up in microscopy workflows that are established sort of in an ad hoc manner, and then they become difficult to share among laboratories, because that was never the purpose, right? My purpose is just to answer my scientific question. That was primary goal. Once I have answered the scientific question, perhaps there is not so much incentive for that group to necessarily package this in such a way that can be easily shareable, right? Although I will also admit that there has been a lot of effort in the community to try to actually work on this. So this is a question that the academic part is also trying to answer. At the same time, of course, the commercial sector is offering more and more solutions, right? So now you will find image analysis, you will find artificial intelligence, all of these workflows, they are all offering it up, which is great, but also creates sometimes fragmentation of the market, which means that just because I can do something in one particular company and I'm confident that maybe the next time a user in another place is asking me for help, but that software is completely different, then I might have problems translating from one place to the other to be able to share this workflow, right? So that's where we believe at the Center for Cellular Imaging that microscopy core facilities are key players in this field, right? So that we could try to then work on this, try to take these methods and concepts and make them available to the research community, right? So I think that we can take some of that load from the research groups and say, okay, we should be perhaps, at least in my mind, some of the ones that are responsible for making this more available. Now, in this sense, we are very happy that our efforts on these lines were recently recognized by the SSF, which is the strategic funding for research in Sweden, who is now supporting the development of smart microscopy at the facility via our research infrastructure fellow grant. And that being said, I'm also happy to tell you that we are recruiting. So if you are here in the room or in the crowd and you find this very, very interesting and you would actually like to now access this challenge from a core facility point of view, please let us know afterwards. I would be very happy to talk to you. Now, going back to what I just mentioned about the smart microscopy and industry. So these imaging workflows are, of course, not an endeavor of only academic groups. Now the microscopy industry at large is jumping in, right? So what I would love to do now that I have the opportunity of having the industry board here is that, well, what I would like to do is see if we can help to align the needs of users, core facilities, and microscopy companies in such a way that we have a bit of a roadmap, right? I know that this is a very challenging task, but maybe there's some things that we can agree on so that we can kind of, come to a successful, not only meeting, but future of this field, right? So with that, I'm again very thankful for the invitation, and let's see how this works out. So that was kind of like the general introduction to the topic. Again, if you have any questions, just please let me know, right? So now what I will show is just some general application examples. This is just to sort of illustrate some of the possibilities, and then I'm taking them from some of these pilot studies that we're doing at the facility. So I think one of the most intuitive or obvious things that become is this idea of targeted imaging, right? So the idea is that in the field of image analysis, we have been using image segmentation and object detection for many years, but usually in the purpose of quantification, right? The images have already been taken, then we analyze them and we try to extract some information from them. In the context of microscopy now, we're gonna do this object detection, but actually as a guiding step, right? So it's not necessarily for the purpose of analysis, but now we want to find, for example, if we use a model for object detection, we might be interested to find a cell that is in a specific cell cycle, and only then there is where I'm going to do my next step. right? Now, what I show you here is a project which we are currently working on, and I think our friends from Aquifer also have been working on similar things, which is the idea that, okay, now we are doing a zebrafish embryo multi-well assay, and we want to go to each well. We also want to then identify morphologically certain cues from the fish, and then go, for example, to the brain, and we want to image some of the neurons. Not only that, what we envision is that we also want to adapt our workflow to not only the morphology of the fish, but also to the intensity and shape of the neurons. So that's kind of what we're working on. And then what you might imagine is that one of the challenges is that even if the fish is sedated, sometimes it moves and it's very light sensitive. So actually, the more light I put on it, the fish might jump. So all of these things are things that we have to consider while we are working on this workflow, and that makes it very, very interesting also from, let's say, the difference between targeted imaging on a fixed sample versus a live sample. Now, the previous example was, okay, we are finding objects, and depending on what we find, we might change the experiment. Now, the next step is going to be, okay, now that I find my objects, I want to follow those objects over time, right? And in there, of course, one of the, what you see on the top right is a beautiful example coming from TrackMate, right? So in a classical sense, this is what we would do. We would just have the microscope sitting on a particular field. The cells will move, and then with the image analysis, we can follow these cells, right? And we can get information about them. Now, what we want to do is that we want to also use these strategies of single particle tracking on workflows that will adapt to, for example, where the cells are moving. Perhaps there is a subset of cells that I want to follow, so the field of the microscope adjusts as the cells are migrating. Then what I'm showing at the bottom now, for example, is a very interesting project where we're monitoring goblet cells in a gut organoid. So those are the cells that are loaded with this thing which is called mucin. That's the mucus, right? So when there is somehow, if your gut feels that there is some kind of attack, they might just release all of this mucin to sort of clean it out. So one of the big challenges there is that in these models, of course, they are three-dimensional, they are large. We have to be able to go and reach them with our microscope, but at the same time, when we trigger the biology that is interesting, these organoids moves a lot, right? They do like fantastic things, and then we have to follow and adjust and you know, try to follow these arms. And at the same time, depending on what we are interested in, we might have to see at different resolution levels. Another thing is that I believe that in this field, this idea of image translation is gonna be huge, right? So I know that many microscopy companies are also offering this now in the image analysis packages, but in principle, this should allow us to also follow live cells for extended periods of time, because now we should decrease phototoxicity if we can translate, for example, a fluorescence field into transmission image into what should be the fluorescence field, right? And then finally, another thing that I believe is very, very exciting nowadays is this idea of image denoising for faster acquisition. So for example, those of you that have read the article Care, that was one of the first things that they show, right? So they were trying to show that they could image much faster because they had these very advanced denoising algorithms. But then the idea is that I believe that also this is one of those things that can help us in fields where we know that the image acquisition is you know, notoriously time consuming. And one of them, which is very close to us at the facility because we are very much invested, is this idea of correlative array tomography, where we have a sample which we have fixed, and then we sort of it's like if you would have a salami and you would slice it, right? So you put all the slices on the surface, and then by sequentially imaging all of these different slices, you can build back your 3D view at extremely high resolution, right? Now, as you might imagine, there's a lot of things here that we can automate. So all of these steps of object detection, target detection, and so on, this is something that we would love to automate. But at the same time, what we would really like to see is that, okay, we want to then do it faster as well, not only automated, but faster by using these denoising methods. And one of the also interesting alternatives could be, could we build in the acquisition pipeline already the acquisition of the data sets that we finally use for the training of the networks, right? So as you know, in some of these methods, it's needed to have pair of images of high signal to noise versus low signal to noise ratio and so on. So it would be extremely useful if we could also start to build all of that thing into the system. So with this idea, so I'm just trying to also show you that we could start with a very simple concept of finding objects, to then link that idea that, okay, I don't only want to find the object, I want to track it over time. And then the last one is really sort of connecting all of these different building blocks, right? It's not only finding the object, it's not only tracking it, it's also using a lot of different image analysis tools inside. So what happens is that the workflow design for such methods can become very, very challenging, right? And the reality is that, at least in my experience, this is an extreme balancing act, right? So you really have to see how you manage to make such pipeline available. And there is no a clear roadmap that I can come and tell you, if you follow steps A to B, you will be successful, right? So I think that there is some general considerations that I think we can, I hope we can all agree on. And well, the first of all, in my mind, is of course the safety of the equipment, right? So you see this beautiful video. I don't know if you follow that on Twitter, but that's great. These microscope nightmares. So, you know, that is my nightmare. That I'm going to program my microscope. I'm going to make a mistake and I'm going to ram the objective into the sample, right? So, and that is a huge worry for all of us working on automated microscopy. I see some smiling faces. I think this is not my own nightmare only for me. So, of course, the first thing that we should try to do, okay, how can we build safety into the microscope in such a way that I know that during the whole thing, when I'm testing it, I'm not going to break my microscope, right? So I would say that's consideration number one. Consideration number two, which is very important, is to have test samples that are easy to prepare for the workflow, right? Unfortunately, a lot of the real biology that we want to study are samples that might take weeks to prepare, right? That are extremely expensive for the user to do. We don't want to test our protocols on that. So we have to find strategies into, okay, what is the simplest, most basic test sample that I can use to make sure that the workflow is working. And in here, there is a very interesting tool and option, which is this idea of hardware simulation. So if we would manage to simulate our hardware, there's a lot of kind of software tools that we could already test, right? So am I communicating well? If I say move the motor, will the motor move? So all of these things that have nothing to do with the sample, we could already try to test in these simulations. And then we know that at least that part is working, right? And then where I'm going to spend a little bit more time after this slide is this concept of divide and conquer, which I think is for so far the thing that has really worked for me, and we're going to cover that in a minute. Now, in the next session, we are also going to cover this idea of the communication speed that might be an issue. Of course, if the sample is fixed and we have all the time in the world, that's kind of, let's say, relatively easy. But if it's a live experiment, then we need to make sure that the round trip of, of the feedback is fast enough for us to do what we want. And the final thing that we'll cover in session 3 will be then the data management of image analysis. Okay, so what is this idea of divide and conquer? So this is this comes from the programming world. At least that's how I got introduced to that. And I think it's a concept that really nicely applies to smart microscopy. And the idea is that you might imagine, okay, and I think we do this intuitively every day of our life. So if we have a very, very big problem, sometimes the solution is not to solve that problem, but to try to break it down, right? So we're going to say, okay, I'm first going to identify small subsets of problems. And the idea is that we should do it in such a relatively smart way that if we were to solve each subset of the problems, once we connect them all together, we have found the solution to the big question. Now, what I find very interesting about this divide, step, conquer, and combination is that if we do this, again, in a sort of smart way, each sub-problem is very well defined. Therefore, it has a very specific input and output, right? So then I could create a mock sample just to test that unit, right? So I don't have to test the whole workflow. I just need to find a mock sample that helps me to test one of these sub-blocks. Now, if this sub-block is successful, then it kind of becomes, again, I'm sorry, I love Lego. That's why I bring this analogy all the time. It brings like a little Lego block that now you have under your belt, right? So this is part of your tool set now. If you need it for a different user in a different context, you can bring it up, and hopefully you don't need to tweak it so much because you have worked very well on this subunit, right? Now, this is also where I think perhaps is where we could get help from our industry partners, which is that I think that from your perspective, it's going to be very difficult to guess a full workflow, like what the researchers are going to want to do in the future. It's almost impossible for you to know. But at least if we can identify some subunits that then you can say, okay, if you will work with us, we can help you with this step, and that step, then you just have to solve the other kind of things around it. I think that would be quite powerful. Also, I think that this allows for a concept which at least that's how we are tackling this issue right now, which is this idea of the minimal viable product. So what I try to offer to the researchers when they come and they're interested in, first I tell them, okay, this is going to be very challenging. It's going to be very exciting as well, but this will take time. So what I would like to do is to provide to you a version of the workflow which has just enough features that solve some of your current problems, right? And then the idea, okay, I will help you to step, let's say B, which is very time consuming for you right now. Still, the whole thing is not automated, but I give you enough that you are excited to come to the lab and try it on, right? So you come to the lab, you try that sort of minimal version of the product, and then you provide me feedback and you provide me data and you provide me with a lot of useful information that I can later use to then continue with the future development. So I think this is indeed the strategy that I would suggest to tackle these questions if you are, for example, in an environment such as mine, which is a research infrastructure, right? So again, with that very quick view, then I would like to finish this introductory session. I would like to, of course, again, thanks to all my colleagues and also to the founding bodies that helped me to be here. And again, I guide you to our website. So please go there if you are interested in our services. And if you have any questions, then it would be great. I think we have a few minutes to cover some topics now, or perhaps we can save this time just to have a longer question at the end of the day. So with that, thank you very much. And well, again, thank you for the invitation and have a great day.